pandemic. I hope you guys are all fine. And I'm going to talk to you a little bit about our work, which is about the pathogenesis of Zika virus during cortical development. Everyone hears me okay, or should I speak louder? Is it okay, Attilio?
Yes, I think it's actually very clear.
Okay. So yes, I've been developing this work at the Federal University of Rio de Janeiro. And first. Okay, so our work is about the cortical development, and we are very interested in the early aspects of cortical development. And the early aspects, the most important early aspect of cortical development are proliferation, where the progenitor cells divide in the progenitor layer. Don't know if you can see my cursor here, if I point to the progenitor layers.
Yes, yes.
You can see? Yes, okay, great. So yeah, so in the early development, the progenitors give rise to the neurons, who then perform neuronal migration and establish themselves in the cortical layers. So after neuronal migration, they differentiate and then they extend axons and dendrites to connect with each other. And why these are very important aspects of cortical development? Well, because if there are any abnormalities in those aspects, so that will generate malformations. So we can group those abnormalities in 3. And we can say that there is the group 1, which is related to malformations caused by abnormal proliferation or abnormal cell death, so you can give rise to microcephaly, so smaller brains. The second group of malformations is related to abnormal neuronal migration. For instance, here you have a double cortex, so you have a defect of neuronal migration that will generate a disorganization of the brain and can give rise to, for instance, to lissencephaly, to smooth brains. And the third type of group of malformations can be related to aberrant connectivity. So for instance, when you have a not functional abnormality between the 2 hemispheres, like when you don't have the corpus callosum, for instance. I'm going to talk more about the corpus callosum in a second. But the human development is very complex. And obviously we are very interested in those details of early neurogenesis and which type of progenitors that will generate later the neurons, but also after the phases of neuronal migration, neurogenesis, neuronal migration, we have the establishment of the early connections. And here I will introduce a little bit about the development of corpus callosum. So the corpus callosum is a structure that connects, formed by axons, that connects the 2 hemispheres and mainly connects most of the neocortex. So here you have a scheme of a coronal section. So if you take a most anterior part of the brain and cut and look in the microscope, what you can see here, represented here in red, you have a neuron that extends its axons through the midline, through the corpus callosum, and then connects to the other hemisphere, to the other cortical hemisphere. And this early connection is formed because there are a number of molecular signals in this path that signalize the axons where and guide the axon where to grow. So here you can see also a representation of those glial populations. For instance, here you can see the midline glia, the inducible glia, and the glial wedge. So those populations are very important to release molecules, attracting molecules or repellent molecules as well, that will guide these axons through the corpus callosum. So I'm talking about these populations because I will later mention about them as well. So this happens during the development, and interestingly, the same molecules can also guide axons, also guide blood vessel development, which is another aspect of the corticogenesis. So the blood vessels, they develop during corticogenesis concomitantly with neurogenesis through the maturation of the brain. So you can see here a representation of this schematic representation of a coronal section, and you have here the progenitors, and you can see there are two plexi being formed during brain development. You have the periventricular plexi here, very close to the ventricular zone, and also another plexi that comes from the pia and then invade the cortex and then start branching. So these early aspects of development are crucial for the correct formation of the brain. And in this talk, I'm going to talk about two regions of the cortex. I'm going to talk about the neocortex, and also I'm going to talk about the hippocampus, more specifically the dendrogyrus. So the hippocampus is phylogenetically older and more primitive than the neocortex. But I will exemplify these two regions. And obviously, we are trying to understand here the human brain development. And you can see here you have a scale of when the events happen in the different weeks of gestation of the human brain for the cortex and for the dendrogyrus, these two regions of the cerebral cortex that we will talk about today. But we use, mostly in this presentation, I use human cells or mouse brain in vivo experiments. So this is possible because we know that those events, they happen in the mouse in a similar way. So you can also compare these two events, like for instance, which is the period that happens neurogenesis, migration, or maturation in the cortex or the dendrogyrus. So this information is very important for us to model human diseases. And the human disease that we are very interested here is the congenital Zika syndrome. So the congenital Zika syndrome was first identified as a abnormality related to the microcephaly, to the reduction of the brain. That happened first in Brazil in 2015. But we also learned that there are other abnormalities, that microcephaly is really the tip of the iceberg, the most apparent abnormality, because in fact, these patients, they also have corpus callosogenesis, when they have this big pathway that are malformed. You have lysencephaly, also ventriculomegaly, when you have these enlarged ventricles. You can see here, and you can see also a lot of cell death, among other malformations. So this mainly happened in end of 2015, starting 2016 in Brazil, when we noticed that there was an explosion of cases of microcephaly, first in the northeast of Brazil, and then later in other regions. And these microcephaly cases were correlated to symptoms of Zika, which was a virus that was recently arrived here in Brazil. And at that point, we didn't know much about Zika virus, only that it was from the same family as dengue virus, yellow fever. So it's a flavivirus that has mainly expresses 10 proteins, 3 structural and 7 non-structural proteins. And we also knew that the Zika virus had actually 2 strains, one in Africa that never left Africa, an African strain, and another one which suffered a mutation and gained the Asiatic part of the world, and then also populated. Also, this strain is now spread in the whole, all the continents in the whole world, mainly in the tropical areas here in red. So we know that Zika virus is transmitted by mosquito bites from the Aedes aegypti and Aedes africanus as well. And we also know that Zika virus is transmitted by sexual activity, and it can stay in semen and can be transmitted, it can be found in the semen, be transmitted after 6 months of the first infection of the patient. So it really scared most of the non-tropical countries, and it is indeed a danger because it causes the brain abnormalities that I've talked to you about. And our goal here is to understand the pathogenesis of Zika virus. And we first started by understanding how Zika would influence the proliferation or the survival of the neuroprogenitors. And we did that in two approaches, one in vitro with 2Ds and 3D cultures, and the other approach was in vivo with to understand different developmental stages with different animal models that I will explain to you. So we first used Zika virus coming from, isolated from monkeys, which is the 766 line, you can see here. And also we were very lucky to collaborate with many virologists that had established the Zika virus from Brazilian patients. And so the first experiment was done with the African Zika virus, and the later experiments were done with the Asian strain, which is the Brazilian isolated strain. And what we first meant to understand here was if Zika was actually causing microcephaly, because at that point we didn't know really, and how Zika acted. So this is very important to understand the pathogenesis. So we exposed the Zika virus to a neuroprogenitor culture. So we exposed Zika virus here in red to the SOX2 neuroprogenital cultures, and we can see that these cultures, they get infected and they replicate. So if we collect the supernatant of these cultures in 2 days and compare the RNA production with the 3 days, you can see there is an increase. So these neuroprogenitor cells, they are actively replicating. They can be infected and they can actively replicate the virus. So then we move to a little bit more complex model, which is a 3D model we call neurospheres. So these neurospheres, they are derived by neurostem cells, SOX2 positive. So the goal here was to understand how Zika interferes in the proliferation and also in the survival of these neuroprogenitors in this 3D system. So here you can see after 3 days, if we expose these neuroprogenitors to agitation and differentiation media, you can acquire this neurospheres, these spheres full of cells such as nestin positive progenitors, SOX2, but also S100 beta and 2G1, which is a marker for neurons. So you can have the neuroprogenitors giving rise to glia and neurons very quickly. So in a week, you can already assess proliferation and also survival. And by electronic microscopy, you can see here that we could find the Zika virus in those neurospheres, and that was actually performed by Dr. Rodrigo Madero. So after 3 days exposing the neurospheres with Zika, we could see very fast, only after 3 days of infection, that the morphology of these neurospheres were completely aberrant. So instead of being brown, it was completely aberrant with lots of cell deaths, lots of cell death. After 6 days, this culture was completely absent in comparison to the mock. Mock here is just a supernatant of the virus cells that we cultivate the Zika virus. That we actually, we use the virus cells to replicate and then to isolate the Zika virus. So if we use only the supernatant of beta cells, that's our control. You can see here you have on average 250 neurospheres after 6 days, but then in different concentrations of the virus, you can see that the culture is completely depleted. Now, this was done with the African Zika virus. We also performed the same experiment with the Brazilian Zika virus. And interestingly, you can see here that we have the same phenotype, that we have a lot of cell death, caspase positive. We have also labeling for Zika. This is a quantification. But interestingly, the kinetics of this infection is different. So it's slower, it's 2 times slower than the African Zika virus. So as you can see here, it takes 12 days for the neurospheres completely be absent in this culture with Zika virus, with the Brazilian Zika virus. Now we asked what were the molecular effects of Zika virus, and for that we did 2 techniques. We did proteomics with Dr. Juliana in Unicamp, and we also sent the samples of these neurospheres after 3 days to Dr. Janaína and the Instituto Evandro Chagas. And then we collected the list of proteins, the deregulated proteins and deregulated genes, and we combine it to create an interactome map. And we had more than 300 genes and proteins deregulated. But interestingly, what we can understand from this map, which is quite complex, I will guide you here a little. So in red, we have upregulated, and in green, we have downregulation. So in these symbols, you have the genes, and the round ones, you have the proteins. It's just a representation of upregulated genes or proteins together in this list. And interestingly, what you can see that you have deregulation of viral replication genes and proteins, and that's normally upregulated. So you have upregulation of viral replication here in red, but you also have upregulation of genes and proteins related to DNA damage. And interestingly, the most deregulated process in the cell was the cell cycle progression. So what we could see was an arrest of cell cycle. And obviously, if you have those cells which were generating the neurons arrested, the outcome was that you have a downregulation of the neurogenic program. So just as an example, you have downregulation of SATV2, which is a transcription factor important for the callosal population. You have also downregulation of NeuroD1, which is a master regulator. So it can actually, it's a transcription factor that can actually activate a package, a neuronal package for the neuronal differentiation. So what we concluded here is that the way that Zika virus was acting in the neuroprogenitors was actually these neuroprogenitors were being hijacked and arrested in order for the virus to be replicated. And obviously we had to confirm these targets. So by different techniques, by immunofluorescence, like all this, from these 300 genes that were deregulated, we confirmed the expression of some of genes, for instance, toll-like receptor 4, which is an important gene for the innate immunity, and also DDX6, which is an important gene for the replication of the virus helicase. And we went further to actually connect the causality of Zika with the brain, with microcephaly, with this brain abnormality that causes the reduction of the brain by using cortical brain organoids. So the brain organoids, it's a technique that has been developed in 2013 to study, obviously, brain development. And interestingly, Madeleine Lancaster, the first author of this paper, she actually used to validate her amazing technique, microcephaly, because microcephaly, it's a malformation that it's not entirely modeled. It's modeled by, let me rephrase, microcephaly can be modeled in vivo with animal models. So you can see here, and you have genetic forms of microcephaly when you have mutations, for instance, the microcephalic gene, and you have the knockout of the 2 copies of the gene, you have a reduction of the brain. This is in the mouse. You can see the knockout mouse has a reduction of the brain in comparison to the wild type, but this reduction is quite subtle and doesn't reflect the fold reduction of the human. As you can see here, this is an example of microcephaly in humans in comparison to a patient, in comparison to a control, which is much more severe. So with this technique, with the brain organoids, very interesting because in vitro, we can recapitulate aspects of human brain development, such as the early aspects, such as neurogenesis, neuron migration, and the morphology as well of the brain. And as you can see here, as Madeline Lancaster showed with human cells from patients, microcephalic patients, compared to controls, you can see you have a smaller brain organoid. And here you have the ventricular zone, and you have a smaller, let's say, cortical wall. So we collaborated with the group of Stephen Regan, which was very important because they had already this technique in place, and they had showed previously that they could reproduce the Lancaster protocol. And we exposed those brain organoids to Zika virus. And as you can see here, there's a reduction of 40% of the area of these brain organoids. So we thought, okay, so Zika can cause this shrinkage of the brain organoid, which is a macrocephalic model in vitro. But is this a feature of any flavivirus? If we expose the brain organoid to any flavivirus, would we have the same result? And the answer was that we exposed, for instance, the neuroprogenitor cells, the SOX2 positive, to dengue virus type 2, which is also neurotropic. You can see there is infection of those cells, but the result of, for instance, in terms of caspase positive, you can see in comparison to Zika virus, dengue virus doesn't kill as much as Zika virus. It actually, it's very similar to the controls. And when we expose, for instance, the neurospheres to dengue virus, we don't see the same result as in Zika virus. And similarly, dengue virus doesn't shrink the brain organoid as Zika virus. So it seems to be a very specific feature of Zika virus. So the effect of Zika virus in infecting the neurostem cells, causing cell death, arresting the cell cycle, and modifying, altering, impairing the brain, the neurospheres morphology, and also the growth of these brain organoids. We also saw that there was an upregulation of viral infection targets in the Zika infected neurospheres, DNA damage, and we have a downregulation of cell cycle progression and neuronal differentiation targets. But meanwhile that we were doing this research in vitro, lots of research has been developed in vivo, and that was very important because one of the questions was how Zika enters in those cells. And the idea was that there were receptors that could facilitate Zika to enter in specific cells. And this receptor, AXL, was very characterized in the brain development. So AXL is a receptor, tyrosine kinase receptor, here in green. So you can see that it's labeling, so again, the same coronal section. You can see here the ventricular zones. This is in the human brain. And you can see here, you have in green, you have AXL in the ventricular zone and also colocalizing with SOX2, which is a marker for the progenitors. In the outer radioglia, it's very important for the expansion of the brain, the human brain, also labeling in green with AXL. But interestingly, AXL was also labeled in the blood vessels. And yeah, so angiogenesis is an aspect of brain development, very important for regulating the blood vessels formation, but also regulating neurogenesis. But to study angiogenesis, we could not study in the brain organoids because the brain organoids have this limitation that you cannot study angiogenesis or the brain vessel formation. But for that, we had to use the in vivo models. And at that time, in 2016, there were 4 papers coming out at the same time showing models of brain development and Zika virus infection. So one which was very important was this paper was out in Nature, showing that you can, if you infect with Zika virus, some animal models, so the immunocompromised ones, you can see that the whole animal is smaller, but also the wall of the cortical wall is smaller. And also another technique to infect brain development, if you infect directly in utero, you can also have the same model of microcephaly. Another model which was very important was this one, which is blocking the interferon receptor 1, type 1, or using a knockout animal, which is the interferon receptor 1 knockout. So if you infect via either subcutaneously, intravenously, intraperitoneally, these animals are immunocompromised, and you can actually model the congenital Zika syndrome. So the congenital Zika syndrome was not modeled by immunocompetent animals. And this paper which combined the 2 techniques. So we embarked in this, in designing an animal model, and we used here a interferon gamma knockout. So this was done in collaboration with Professor Maria Bellio, who had already these animals, and that was really important for this work. So we used those animals which are immunocompromised, type 2 interferon knockouts, and then we injected intravenously the Zika virus in the stage of E12, which is a stage of the development that you have a peak of neurogenesis. And as you can see here, the Zika virus 3, so this one is a cortical coronal section, and you can see here that this brain is smaller, and the cortical wall is in fact smaller. And you have the cortical thickness reduced, you have reduction of the cortical progenitors in P2, postnatal day 2, you have reduction of the infragranular layers and the supragranular layers, here the infragranular layers and the supragranular layers. We also had to characterize this model. So we had to detect the Zika virus RNA by qPCR in maternal tissues, but also in the pups, in the offspring of those mothers. So we infected in the pregnant mother in the day 12 of the embryonic day 12, and then we collected in P2. We also collected after 3 days after infection. So, and as you can see here, this is the skull. There is a reduction of the skull and also a reduction of the overall the brain. So you have a lighter brain and a smaller brain. And this recapitulates pretty much, it's very consistent and recapitulate pretty much what we can get in terms of microcephaly in mouse models. So one question that we had, if we wanted to understand how Zika could affect the brain bloody vessels development, was if these vessels, these cells were actually infected or colocalized with Zika markers. So we had here IB4, which is an isolectin, also labels microglia and blood vessels. So you can see here that it's colocalized with Zika virus, and with J2, which is an antibody that marks RNA, double-stranded RNA, and you can see here there's a colocalization with the J2 and IB4 in the cerebral cortex, and not obviously in the mock. Here's another representation of this IB4 and Zika virus, the blood vessels in the brain and also in the placenta. So in this paper, we also describe that possibly Zika virus was infecting the placenta blood vessels and entering as well via the blood vessels. So our question was if these infected animals, they had any abnormalities in in the blood vessel development. So for that, we looked at IB4, which is a marker for the blood vessels. And as you can see here, only after 3 days of infection, so we infected in E12 and then in E15, you have the number of vessels were the same, but the number of branches of those vessels were reduced, and also the length of those branches. So you have the length, these vessels without or with less or shorter, with less branches and shorter branches. And so overall, the area of the blood vessels area was reduced. So we're not sure if there was any, this was related to cell death of these cells, or the death of these cells, or a defect in proliferation, or actually it was a problem with differentiation of these cells. So we colocalized here with nestin. So laminin is a marker for the blood vessels, and nestin for progenitors. And as you can see, there is a reduced amount of lower reduction in the colocalization of those markers. And here with other markers, with laminin, CD31, you can see clearly there is a reduction of those markers. And if we looked in the later stage of the development in P2, you can see that though some of the parameters are normalized. So it seems that there was only a delay in the development. So for instance, the number of branches already catched up, and we still had the defect of the length of the branches. So this is after P2. So in order to understand a little bit more molecularly how Zika was actually interfering in the blood vessels development, we performed a proteomics. So this was not the wide proteomic that we did in the neospheres. We actually had a panel of some proteins that we could check, and these proteins were related to angiogenesis, and we could check if they were deregulated. And as you can see here, there's a lot of deregulation, but mainly upregulation of some of the targets, and all these targets are related to angiogenesis, but also affects, for instance, neurogenesis and overall the brain development. So as a conclusion, we can say that yes, the Zika virus affects brain vessels development, and this could also implicate, obviously, because angiogenesis has a crosstalk with neurogenesis. We can say that this could also be another factor that can reduce neurogenesis. So we know that Zika virus infect directly the progenitors, but what we are saying here that perhaps with a delay in the angiogenesis, you could also affect neurogenesis. And this has to be checked yet. But what we saw in terms of neurogenesis, if we look again in these coronal sections, if we zoom in the ventricular zone, and you can see here in the ventricular zone, you have aligned the pH3, which are the marker for mitosis. So you have mitosis here in the ventricular zone and mitotic cells here in the subventricular zone. When we look at and we counted, quantified the amount of cells of pH3 cells in the ventricular zone and subventricular zone of the infected animals 3 days after infection, you can see here there is a reduction of these cells in the ventricular zone and in the subventricular zone. So it seems to have altered the cell cycle of both progenitors. And this obviously has a reflection in neurogenesis. So at this moment in E12, this CT2 population in the cortex is generated. So is actually being generated. So after 3 days, we can have a look and quantify the amount of CT2 numbers, the number of cells in an area. And we can see there is a reduction of those cells after 3 days. So neurogenesis being affected, as well, obviously, an affection. There is also an impact in the progenitors. Here is the PAC6 positive cells in the ventricular zone also reduced. So in terms of proliferation or cellularity in the brain, we could see that there was the neurogenesis being affected, but also interestingly, the Zika virus is upregulating or is increasing the number of microglia. So after 5 days of infection, you can see an invasion of microglia, and this is labeling the microglia IBA1 marker. And you can see there is a upregulation, either a proliferation of this microglia or also an attraction for the infected area. And this is very marked. And at the same time, 8 days later, you have an increased amount of caspase. So here in green, you have cell death in the cerebral cortex, very strong, showing that there are lots of cell deaths, lots of cell death. This is in the cortex, in the neocortex. When we look at the dendrigyrus, another area of the cerebral cortex, we can see here that infecting P0, which is the peak of neurogenesis in the dendrigyrus, we can see it with the Ki67 marker, which is a cell cycle marker. You can see in 3 days after infection, 7 days, and 14 days, you have labeling here proliferation, and this is severely altered in the Zika virus infection. So when we infect those brains with Zika virus, you can see there is a severe reduction of the proliferation in those dendrigyrus. Now, at the same time that you have a reduction of proliferation, there is an increase of cell death. So here we label cell death with caspase, activated caspase 3, and you can see an upregulation after 7 days of infection. And there is a reduction as well in neurons. So here we label neurons in green, NEUN, which is a marker for neuronal marker. So you can see here the mock showing the dendrigyrus here, the granular cells, and then you have a reduction of those cells in the Zika brains. So you have a disruption of the cell cycle, an increase of cell death, and also reduction of neurogenesis. So then we decided to look for another aspect of the brain development, which was the organization, the migration, the distribution of those cells. And we started doing this by looking at the progenitors. So again, we have a coronal section and we gave a pulse of EDU. So this is an analog of timidine, and we give 2 hours pulse. So it actually being incorporated in phase S, in S phase. So we divide this proliferative area in beams. So you have these beams of 20 micrometers, and then we can count these cells. And as you can see here, this is the control, and this in the affected animals with our congenital infection model. So after 3 days, as you can see here, you have a different distribution of those cells. So these cells, they perform a interkinetic nuclear migration. So their nuclei is here in the basal levels when they are synthesizing the DNA. So labeling with EDU here with 2 hours, and then they perform this migration to divide here in the ventricular zone. And as you can see here, there is a wrong distribution, altered abnormality of the distribution of these progenitors. Now, when Raisa, a PhD student in the lab, looked for the distribution of neurons, for instance, in the brain, after 3 days, same time as the progenitor that we saw this impairment in the distribution, here, after 3 days, Raisa could not see any difference in the, actually when she tried to quantify the distribution of those cells and this used SATP2, which is a marker for callosal cells, she didn't find any abnormalities in the distribution, although if you infect the cells in E15, you have a reduction of SATP2, because that's the moment that those cells are being generated. So you have a reduction of SATP2, but they are supposed to be. At least at 3 days, they are where they're supposed to be. Interestingly, if she performs a later infection, she can see a much different scenario. So here, infecting P0 and leaving 11 days. So you can see there is a reduction in the cortical wall. Again, this is a coronal section. There is a disorganization of the cells, of the layers, as depicted here. So here is the mock. This is the Zika. Sorry, I didn't put here mock Zika, this comparison, mock Zika and mock Zika. Here, as you can see, she also saw a disorganization in the hippocampus, mainly in CA3, but also in other areas like the dentigyres. But coming back to the cortex, when she looked at the CTIP2, which is another marker for projection neurons, she could see a disorganization this time after longer exposure of the virus. So normally those CTIP2 positive cells, which are labeled here in green, are in the lower, in the infragranular layers. But here you can see the Zika virus, and she did a higher magnification here. And as you can see, you have the CTIP2 positive cells in the upper and the supragranular layers, which are not aberrantly found, so ectopically. Braisa also examined other types of neurons, for instance, the interneurons, and she looked at the carbindine, I don't know, these carbindine neurons, interneurons, and she could see that they were also disorganized. Mainly there was a reduction in the carbindine neurons, and mainly they were lacking in the infragranular layers. So as you can see in the mock, they are distributed, but then in the Zika virus, they were lacking here in the infragranular layers. And another interneuron subtype is somatostatin, and you have also a reduction of those populations. So then we went on to examine after the generation, after the proliferation of the progenitors, after the organization and the migration of those cells, we went on to examine morphology of those neurons and differentiation. And for that, first, we look at MAP2. Oh dear, what happened here? Sorry, I don't know what I did. Okay, let's try it again.
You need to share screen again.
Okay. Share screen. Sorry, my keynote closed unexpectedly, so bear with me. In the last slides, just to wake you up.
Don't worry, I hope it's not a problem with my subscription to Zoom. It was renewed yesterday, so I hope it's not.
Okay, so no, I think it was here, the problem. Zoom. Okay, can you see my screen now?
Yes.
Right. So as I was saying, we looked now for the maturation or the morphology of those neurons upon Zika infection. And first, we look at MAP2. So MAP2 is a microtubule regulating protein, which is labeling mainly the dendrites. And we can see the labeling is very different. Oh dear, again, I don't know what's wrong here. I'm so sorry. It's my keynote is crashing.
Don't worry, it happens.
Let's start again. Okay. Okay, so we look at MAP2. So MAP2 is a protein which is mainly expressed in the dendrites. And when we look at the Zika virus infected animals after 11 days, and you can see here that the MAP2 is very apparently expressed. So it's not polarized and radially polarized as in the control. Oh, I don't know.
Patricia, maybe you can try without doing the full screen. Just try to.
Yes, I'll do that.
That's
without the full screen.
Let's see if it works.
You can see now?
Yes, yes.
Okay. So this is not the full screen, but I hope you can appreciate the results. Yeah, so the MAP2 was aberrantly expressed. So Raisa again went to look for the neuronal morphology with a very traditional technique. It's a Golgi staining. So it's a technique which was developed by Camillo Golgi, got the Nobel Prize. So it's an ancient technique, but very beautifully labels the neuron, the axons, and the dendrites. And as you can see here, Raisa could stain the control brains and the infected brains. And she could also reconstruct the arborization of the dendrites, the basal dendrites, the apical dendrites, and she mainly found a defect in the basal dendrites. So here is the mark, which is the control, and you can see here with the infected ones, and you have shorter, the length of those basal dendrites are affected. Now we went on to examine if these morphology was also altered in vitro, because in vitro we can isolate better the neurons and we can study better. That was the task of Jefferson. So Jefferson did a cell culture of, so basically Jefferson took a E14, so day 14, embryonic day 14 in the mouse, and then it dissociated those cerebral cortexes and exposed those neurons to a differentiation media. So they were all neurons, so they're pure neurons culture, and labeled for specifically SATP2, which is a transcription factor that labels corpus callosum, and expose those cultures to Zika virus. And you can see here that there is a decrease in when Jefferson measured the length of those axons, and then also later he labeled the axon to see if the axon was the axon, if you know what I mean. And you can see there is a reduction of this length of the axon. And what Tailani found later was that the corpus callosum was actually, the area and the volume of the corpus callosum was actually decreased. So if she infects in P0 and harvests in P7, you can see that the corpus callosum here in yellow, so right in the midline, you can see that this area is reduced. So here is the Zika brain and coronal section, here is the mock. Now what research did as well was to zoom in the midline, and she could label with L1CAN, which is a marker for axons. So it's a molecule that labels axons. So you can see here that you can have the extension of the corpus callosum you can measure. So so she infected in E13, she harvests in E18, and at this moment, you can already see the corpus callosum axons that have crossed mutually, and you have a reduction here in the Zika infected. Now, what she also noticed, if she puts a dye in the brain, so dye is a lipophilic molecule that labels the membranes, and then what she can see and follows those tracts of the axons, she can see some defasciculated axons coming out of the tract that was going to go in towards the other hemisphere. And interestingly, this effect of Zika virus, it could be an effect directly, as Jefferson showed, in the neuron. So the neurons, they had a defect in axonal growth. But also what Raisa found, she investigated the midline. In the midline, you have those glia cells, those populations that are important for guiding these axons. And here in GFAP labels those glia cells in P0 in the brain. And she could see there is a reduction in those type of glia, the endosognesium, the midline zipper glia. So as Zika virus has a tropism for neuroprogenitors and glial cells, perhaps this region was being attacked by Zika virus directly, and then these axons could have been misguided. So this is still under investigation, but what we know is that in P60, so in adult brains, we still have the phenotype of the reduction of the corpus callosum. So the corpus callosum is malformed. You have a reduction even when we normalize by the brain size. But what Taileni could see is that the numbers of SATP2, so the callosal cells, were still there. So the callosal cells have haven't changed numbers, as opposed as another population, the CT2, which is more affected directly. Interestingly, so the callosal cells are there, but you still have a defect in the corpus callosum formation and in the corpus callosum maturation. So because we had this reduction of corpus callosum, also because we had a reduction in the interneurons, we thought that maybe these animals would have a different behavior as well. So first, what Raisa tested, so here was animated, never mind, it's okay. You can still appreciate. So first, what Raisa tested was if those brains were more active. So she used for that a marker, CFOS. So CFOS is a marker that when neurons are depolarizing, so when neurons are active, CFOS is upregulated. And you can see here the Zika brain has more labeling of CFOS, so it's hyper active. And so to test that behaviorally she used a technique that was taught by Professor Henrique Mendonça in FRJ. So she basically performed a seizure test with those animals. So she put these animals in a cage and heated up this cage and measured the time, the latency that those animals start with the seizures. And she could see that the Zika animals, they were more susceptible to the seizures. The latency was shorter, and also with lower temperature, they were already performing the seizures. So just to finalize altogether, what we showed here and what we've been studying in terms of Zika virus pathogenesis is that, first of all, there are good models to study infectious disease in vitro. So for instance, we found a good model to study microcephaly caused by an infection by a pathogen such as Zika virus. And this was very important to connect really what was happening, this correlation of Zika and the microcephaly. As we showed, there was a causation. And we also digged in terms of mechanism, and we found that there were abnormalities in the cell cycle of those cells after infection, and that was halting the neurogenic program. In vivo, we show that the Zika alters many aspects of developments depending on the timing of the infection, and we could see that alters the blood vessels directly as it has the receptors, it infects directly those cells. Also the progenitors directly, reducing the neurogenesis. It also impairs neuronal morphology and connectivity of those brains. So what we predict is that these patients, obviously what we're seeing, we are trying to understand what is the etiology, but what we are seeing is that these patients, they have more susceptibility for seizures, and that perhaps has a root on aberrant cellularity in the brain. So I would like to thank my collaborators very, very much indeed. So we have the collaborators in Instituto in the Stevens Rehan lab, which were fundamental, as well as the collaborators in the UFRJ, Luisa, Paula, and Professor Milka, which were also essential. Without them, we could have not done the experiments. And the collaborators in Oxford University, in the Moners lab with Helen Staub, which is now in the RVC, which also, all these collaborators we helped me and our group to get the grants to fund this research. And I would like to also acknowledge the neuroplasticity lab, which Professor Roberto Lente is the head of the lab, and I'm really, really grateful to be hosted in his lab with my group. Together with this list, lots of people that I mentioned, or if I didn't mention it during the presentation, are listed here, which are also essential for the work. I'm really grateful for all the collaborators, for all the team in the neuroplasticity lab. And here I have a picture of some of them that I mentioned in the presentation. And I also would like to thank you for your attention and for the opportunity, Atilio, to present our work here. Thank you.
Thank you so much, Patricia. Really terrific talk. This was super interesting. All right, so we can open the question session, and I would like to ask everybody to mention the first name and affiliation. I know that there is people from a number of places around the world. So if you could just state your name and affiliation before asking the questions, that would be nice for our records. All right, so.
No? I have a question.
Okay, Bruno.
So my name is Bruno Mota. I'm from UFRJ, but from the Physics Institute. And I was very curious about the microglia becoming more, proliferating more after infection with Zika virus. Can you say something about what they're doing, why they're proliferating more? Are they the ones responsible for eliminating the progenitor cells, or is there some other mechanism?
That's a very good question, Bruno. Thank you so much. Yes, so the microglia is like the macrophages of the brain. So their role is really to be able to remove any pathogen if it exists in the brain. So they are the first one to respond and to proliferate and migrate and go to the site of infection and trigger a inflammation response, or so to say, and they become activated, and they have this morphology of activated microglia. And yes, so we still don't know exactly how microglia acts in terms of the infection of Zika, but what we know, for instance, from the work of Fernanda Barros in adults, is that when the Zika virus infects the brain, the microglia becomes activated, and instead of doing what they were supposed to do, like for instance, they were supposed to be pruning or doing their regulatory activities, they are actually removing the synapses. So obviously that could be accounted for the behavior, like learning behavior defects of those animals, because when you have the removal of synapses, the pruning of the synapses, that is also actually interfering in how the brain learns and how the brain responds to some stimuli. So yeah, so the microglia could be acting, pruning synapses, for instance, because of these deactivation, so this activation of the virus. but in our system, we don't know yet what's happening. But interesting that you mentioned that, because in the midline, remember, let me just come back here. So in the midline, you have a reduction of the corpus callosum, and you have also a reduction of those cells. And here, I didn't show you that, but there is a massive amount of microglia. So it might be the case that Zika is actually infecting these populations, these glia populations, but it also could be the case that microglia is removing those cells, you know, or both. Yeah, so we don't know exactly, but we speculate that yes, it could be actually removing those cells. Yes. Thank
Okay,
you.
I'm asking this because I'm actually very interested in microglia during the proliferative stage, before you start producing neurons, and while still having symmetrical divisions of the progenitor cells. I'm so curious about that because last year I was talking to Stephen Noctor from University of California, Davis. Yeah, I was there, and nice weather when we could still travel. But anyway, so he told me he saw something very interesting in this early stage. He saw that you had these progenitor cells, but that by all accounts are perfectly healthy, and there is microglia that essentially make a tapestry underneath those progenitor cells. And whenever a microglia would touch one of those cells, the cell would die. That's what he told me. Yeah. So could the same mechanism be happening there? It could be that whatever is the Zika virus is doing to the progenitor cells is somehow promoting the microglia murder spree, so that you're losing lots of progenitor cells and therefore have fewer neurons at the end.
Yeah, definitely. Microglia is involved in the removal of those cells. And interesting that you mentioned that, because there was a result that we were very puzzled, which was this one, that we saw a reduction of the progenitors, and we didn't see actually caspase positive in those stages. In 3 days, we don't see caspase positive, you know, and we were like, okay, so maybe there's only a delay in the cell cycle, you know, and then because the cells are not cycling, they're not generating, so they're not, you know, here in this stage, they're asymmetrically generating another progenitor, and one neuron.
They're not dying, they're just not doing anything.
Yeah, so maybe they are arrested here, but also the microglia that we see is a bit later. So the microglia invasion is actually after 5 days. So yeah, it could be a mixture of both.
Cool. That's actually very interesting.
Can I add on to that question? My name is Sheree.
Sure.
I'm a grad student from Princeton. My question is, have you looked at the microglia response that you saw in the knockout mouses with the interferon gamma knockout, I believe?
Yeah, so we saw the same. We saw also an infiltration of microglia and upper. So here it's not it's a picture of in utero infection, but we also have examples of this model here with microglia upregulation.
So you don't think the microglia are bringing in an interferon response, which I guess one could expect, no way?
I think the interferon in this case, because we did the infection, the maternal infection, so the interferon response was very important in this case to be absent in order for the virus to be able to infect the placenta and get into the brain. So that was that's why we use immunocompromised mouse. It's because the interferon response is very important in this maternal environment, you see. And then after, I think the innate response is more of the cells that are being affected here in the brain. And then we have, like, for instance, the toll-like receptor upregulation and so far so on. So yeah, so I think in terms of the interferon response type 2 in this case was very important to be absent in order to have an immunocompromised model.
Patricia.
Cool, thank you.
We have 2 questions from Markley Silva from Heinrich Heine University in Düsseldorf, Germany. There's problems with the mic, so I will just read the question. So do you think the modification could happen onto OSVZ, sorry, I'm not an expert on this, with type C stem cells, apparently explaining the brain malformations, probably linked to astrocyte progenitors, type C TAPs or type A stem cells, that maybe are not capable to recruit blood cells growth?
So can you read it again? Which type? What's the marker that he said? The
Do
type
you think the modification could happen onto SVZ with type C?
SVZ. Okay, yeah. Yeah.
Never
Okay.
heard of this name. Sorry.
So, sorry. So we have not examined the lateral ventricles of the brains in terms of these adult neurogenesis niches. We only examined so far the dentigyres here. But yes, there are some data in the literature showing that in the lateral ventricles, the SVZ, the subventricular zone, is affected as well. So you have a reduction of the neurogenesis, but I'm not sure if it's determined. I haven't seen anything in the literature determining which cell is being affected, if it's the type A, B, or C. So this is still open question, and we haven't examined that. But in the hippocampus, in the dentigyres, Jessica, she was examining this question, and she actually saw a reduction in the SOX2 positive neuroprogenitor cells. So directly in the neuroprogenitor cells. But yeah, so the composition of these two neurogenic niches, it's a bit different. But yeah, so here we can say that the SOX2 positive is affected. I think Jessica also looked for a more intermediate, the TBR2 positive, that are not affected, and the double cortin are affected. So it seems to be a defect in neurogenesis in the neuroprogenitor cells itself.
Patricia, there is a second question from Mark Lee, and is about the SVZ and SCs. So are these more related to the corpus callosum formation? Those cells can easily migrate to the corpus callosum during brain development and can mature as oligodendrocytes and astrocytes in humans and rodents as well. Could be SVZ and SCs the answer for brain repair in Zika microcephaly?
That's a good question. Yes, so indeed the neurostem cells is the root of this microcephaly. So if we could, let's say, inject neurostem cells and we could replace the pool of progenitors, that would be great. And yeah, that would be a great way to avoid the microcephaly. But in fact, another thing, the microcephaly generated, or the cell death, or the lack of neurogenesis of this disease has two aspects. One, which is directly in the neuroprogenitor, and another aspect is what we were talking about, is the inflammation. So the inflammation of the brain, which also leads to a hostile environment and lots of cell death. So mainly there is a reduction of neurogenesis directly because the progenitor cells are not dividing and not giving rise to the neurons. But there is another wave of reducing the neuronal population, which is when the microglia takes place and activates, and then you have a hostile environment, and then you have massive cell death of the neurons. So it would, in a sense, it would help in the first case, but it wouldn't help in the second aspect, because then you have inflammation anyway because of the virus. Yeah, so yeah, it has to be taken into account, these two effects of Zika.
More questions?
I have another question, if that's okay.
Yes.
Yeah.
Could you comment a little bit about how the virus gets to the progenitor cells? And specifically, so is the first particle that the animal comes across, is it replicating before it gets to the progenitor? And if it is, like, for example, if it's being carried via the blood vessels, but the blood vessels are being killed off. Is that kind of like a regulation to prevent it from going to the progenitors? I just don't know about the path of how it gets to that point.
Yeah, so that's a very, very important question. So how does Zika actually get into the brain? Obviously, it's a very, very important question. And one thing that we know is that, for instance, here, it's showing blood vessels in the placenta, and the placenta is infected. The blood vessels of the placenta, the blood vessels are infected by Zika virus. As you can see here, this colocalization. Now, there was a paper, very beautiful paper, showing, it was in Nature Communications last year, showing that monocytes are able to be the carriers of Zika virus. So infected monocytes circulating from the placenta to the brain can carry Zika virus, and then Zika virus can infect the blood vessels and infect the progenitors and so far, so on. So mainly these are the two cell types that can get infected: blood vessels and uroprogenitors, and also microglia. So we see colocalization on these three cell types. We see a little colocalization with a GFAP, which is an astrocyte marker, and we almost, and in the embryonic brain, we don't see a colocalization with neuronal markers. Funny enough, in the adult brain, you can see colocalization with neuronal markers. So we don't know if in the adult brain, these neurons, they upregulate the receptors of Zika, for instance, the AXL or the integrin receptor, which I haven't had time to mention, but it's very interesting for glioblastoma aspects. Yeah. But yeah, so in the developing brain, these are the targets, the cell targets of Zika.
Thank you.
More questions? Patricia, I do have a couple of questions, and I apologize, I might have missed some of the information because I was trying to multitask, so I probably missed a few things and also answers that you gave to some of the questions. So it seems to me that there is at least two defects that contribute to the microcephaly. One is in the neuronal development, and the other one is in angiogenesis, and the two are tightly coupled. My question is,
Yes.
how much the one defect contributes versus the other, which means that, or in other words, is microcephaly due more to defects in angiogenesis and vasculature branching and formation rather than neurons? Because the neuronal death seems to me, or from what you said, and I understood, it's due to prolonged arrest in cell cycle that eventually leads to apoptosis through caspase activation. And
yeah,
this could be due to DNA damage, and you see those proteomic studies showing upregulation of DNA damaged proteins. But it could be either due to really a problem in the neurons, so in DNA damage, which you could monitor, for example, with the anti-H2AX antibody staining that monitors DNA breaks, or it could be indirect. It could be due to the lack of oxygen nutrients that are brought about by the vasculature. So I was intrigued by this interaction between these two systems, these two problems.
I think that's a very, very interesting question to dissect exactly. But to answer your question directly, sorry, excuse me, to answer your question directly, it's clear in the literature, in our work, in the patients, it's clear that the root of microcephaly is indeed the neuroprogenitors, they are the stem cells, because these are the tropism of neurostem cells is higher. So if you compare, for instance, a culture of neurostem cells with a culture of endothelial cells, obviously you have a higher replication and faster replication in the neuroprogenitor cells. And if we think about in terms of microcephaly, microcephaly is a malformation that can be found as well, caused by genetic abnormalities. So you have like the mutation of 2 copies of genes, very important for centrosome formation or for cell division, then this will generate microcephaly. And So the root of those types of microcephaly is the neuroprogenitor defect, is the survival and abnormalities in the cell cycle of those progenitors. On top of that, I think the microcephaly that we see here, it's more complex. So that's why it's a syndrome. It has more impairments in the brain. So you have not only the microcephaly, you have agenesis of corpus callosum, you have lysencephaly, you have a huge ventriculomegaly, and on top of that, you have the angiogenesis defect. But the angiogenesis defect, to be honest, is minor in comparison to the defect in the progenitors. As you can see here, you have a reduction of the length, but you don't have a reduction of the number of vessels, for instance. So it seems to be something very timely. And then after this first stage, it's catched up. It's just a delay. In comparison, when you look at the reduction of the progenitors, this pool of progenitors, they will never be replenished, you know. It's something that it's for life. You have a reduction and that's it.
Thank you. More questions? All right. So I think we can close here. I want to thank you again, Patricia. And I would like to also mention that we will have another talk in 10 days on May the 6th, which will be from Thomas Gregor from Institute Pasteur. And he will talk about transcription in 3D, and in particular, the interaction between enhancer and promoters. And of course, I will send the flyer to everybody tomorrow in the next few days. But once again, thank you so much, Patricia. This was really
Thank
amazing.
you.
It was really, really amazing. And see you.
Thank you so much for the invitation.
See you, everybody, in 10 days then.
Thank you. Thank you so much. Bye-bye.
Bye-bye.